I'm going to be discussing the adolescent stress and neuroplasticity in hamsters article by Craig Ferris. Ferris starts the article off by addressing the limitations and findings of previous aggression stress studies. These studies found that stress during the prenatal period can negatively affect aggressive behavior in numerous mammalian species. Prenatally stressed animals demonstrate arrested development, diminished social skills, erratic and unpredictable aggressive behavior, and increased vulnerability for long-term psychopathology. Ferris found that there is a gap in information on how stress affects adolescent animals, which is important because adolescent animals are confronted with stressful situations that may cause long-term changes in the neurobiology and behavior of these animals. Ferris sought to address this limitation as well as examine how arginine vasopressin and serotonin 5-HT are related to the control of aggression and affected by adolescent stress. From 1988 to 1997, Ferris conducted a series of studies on Syrian golden hamsters. He found that vasopressin fibers are co-localized with V1A receptors in the anterior hypothalamus, and blocking vasopressin V1A receptors inhibits aggression in both resident male hamsters towards small intruders and between hamsters tested in a neutral arena for dominant/subordinate behavior. However, if those V1A receptors were to be activated, aggression in hamsters would be facilitated. Furthermore, if vasopressin neurons localized to the medial supraoptic nucleus and nucleus circularis are lesioned, there is a reduction in agonistic behavior. These results are not only applicable to hamsters, but even in humans, high concentrations of arginine vasopressin facilitates aggression. Similar to vasopressin, serotonin 5-HT receptors are located in the anterior hypothalamus, with a high density of 5-HT receptors overlapping the V1A bonding sites. Now, unlike vasopressin, the serotonin of 5-HT system is associated with the inhibition of aggressive behavior. Ferris' 1996 to 1999 studies found that in hamsters, the usage of fluoxetine, a 5-HT reuptake inhibitor, inhibits vasopressin release in the anterior hypothalamus, blocking offensive aggression of a resident towards an intruder, and suppressing vasopressin facilitated attacks and bites. In summary, vasopressin facilitates aggression, while 5-HT suppresses aggression, in part by inhibiting the activity of the vasopressin system. And these results are transferable to humans as well. Ferris cites that patients with an aggressive personality disorder show a non-responsive 5-HT system and a significant positive correlation between cerebrospinal fluid of concentration of vasopressin as well as aggression. Ferris then goes on to talk about the social behavior of adult animals. In adult animals, it was found that losing fights and being relegated to low social status is very stressful. And adult animals who lose fights exhibit lower levels of testosterone and lower levels of vasopressin in the anterior hypothalamus, lower levels of aggression and increased levels of fear and defensiveness to animals of the same size, higher levels of aggression in smaller sized and equal sized intruders, and temporary conditioned defeat, which is simply submissive behavior. Now, in terms of adolescent animals, the results are slightly different. Ferris utilized studies on adolescent golden hamsters in order to illustrate his findings. A golden hamster is only considered adolescent in a way analogous to humans between postnatal day 25, aka P25, and postnatal day 42, aka P42. It is only between P25 and P42 that hamsters achieve independence from the maternal nest, doubling their weight size, reaching full sexual maturity, as well as reproductive competence. And they also establish social relationships. In a 1988 Delville study, male golden hamsters weaned at P25 were exposed daily to aggressive adults from P28 to P42 and tested for offensive aggression as young adults several days later after the cessation of stress. Here are the results of the study. Adolescent animals with a history of subjugation showed typical conditioned defeat by running from non-aggressive intruders of comparable age and size, which is a result similar to the socially subjugated adult male hamsters. However, when confronted by a smaller, weaker intruder, they were exceedingly aggressive, displaying short attack latencies and a high number of bites compared to their sibling controls that were not subjugated during adolescence. These adolescents demonstrated no difference in basal testosterone concentrations when compared to their control siblings, and they also demonstrated much lower cortisol concentrations than their sibling controls when faced with an aggressive larger animal. They also demonstrated an increase in serotonin 5-HT innervation to the anterior hypothalamus compared to their sibling controls. Because 5-HT decreases aggressive behavior, Ferris concluded that it is possible that the stress of threat and attack in adolescence alters the interaction between vasopressin 5-HT systems, affecting the regulation of aggression and possibly the context-dependent nature of the aggressive response. This idea is demonstrated in Figure 1. Figure 1 depicts the interaction between vasopressin, AVP, and serotonin 5-HT in the regulation of aggressive behavior for adult resident animals without a history of adolescent stress response to an intruder, and adult resident animals with a history of adolescent stress. The first diagram shows adult resident animals without a history of adolescent stress response to an intruder. In this example, the presence of an intruder results in the activation of vasopressin neurons in the medial supraoptic nucleus and nucleus solaris, and the inactivation of 5-HT neurons, removing an inhibitory drive on vasopressin neurons and neurons in the anterior hypothalamus controlling aggression. In summary, adult resident animals without a history of adolescent stress response to an intruder will demonstrate an enhanced aggressive response towards intruders. The second diagram depicts the same interaction, but now between a resident with a history of adolescent stress and threatening and non-threatening intruders. This diagram is also slightly altered to reflect the altered interaction between vasopressin 5-HT systems. In the second diagram, the vasopressin neurons have been made smaller to reflect the decrease in vasopressin concentrations in the anterior hypothalamus due to suppression of vasopressin gene expression. With less release of vasopressin, there is an increase in the density of V1A receptors, which is depicted by the larger receptor. Furthermore, since stress is associated with an activation of 5-HT, the figure demonstrates an increase in density of 5-HT immunoreactive boutons in the anterior hypothalamus to suggest an increased release of this neurochemical signal. With more 5-HT being released, there is a downregulation of 5-HT1A receptors, which is why it is depicted as smaller. For threatening intruders, the presence of an intruder results in the inactivation of vasopressin neurons and the activation of 5-HT neurons, thus suppressing the vasopressin neurons, causing a submissive but non-aggressive response towards intruders. For non-threatening intruders, in this example, the presence of an intruder results in the activation of vasopressin neurons in the medial supraoptic nucleus and nucleus solaris, and the inactivation of 5-HT neurons, removing an inhibitory drive on vasopressin neurons and neurons in the anterior hypothalamus controlling aggression. In summary, adult resident animals with a history of adolescent response to an intruder will demonstrate an enhanced aggressiveness towards non-threatening intruders. These findings have larger implications these findings have larger implications than simply how golden hamsters interact. They also indicate something about the way we humans develop and form behaviors related to aggression and stress. Ferris cites that the American Humane Association reported a high incidence of abuse toward adolescents between the ages of 12 and 17. And this evidence indicates that the incidence of adolescent abuse equals or exceeds that of all other age groups. This is important because a lot of studies pay more attention to the victimization of infants and children without accounting for the behavioral and biological consequences of abuse during adolescence. From this study, it is clear that there is a larger context-dependent nature of aggressive behavior and the neurobiological and behavioral consequences of stress in adolescence. That must be addressed or paid attention to, since there is a highly plastic nervous system in adolescence. Thank you.
Hello, everybody. I'm going to be talking about the Ferris 1997 paper. I think it was really confusing. Don't feel bad if you were confused. It's not you, it's them. They structured it very poorly. Scientists don't know how to write, man. But I'm going to be hopefully walking you through it and kind of creating a more like chronological and kind of top-down approach to this very complex paper. So a little bit of background, we're looking at vasopressin, specifically arginine vasopressin. It's the same thing. It's a diuretic hormone. It's abbreviated in this paper as AVP. We're also looking at serotonin, 5-HT. We're looking at them because previous research has shown that they mediate aggressive behavior. You inject rodents with vasopressin, they become aggressive. You inject them with serotonin, they have a subdued aggressive response. The vasopressin high concentrations are also associated with increased aggression in humans and primates. So let's get on with it. The goals and the hypothesis of this study. So first of all, this study, we're going to be looking at a region of the brain known as the basolateral hypothalamus. Specifically within that, we're going to be looking at the anterior hypothalamus, as this is a region where these two neuron populations interact. So the goal of this paper is to examine the neuroanatomical and neurochemical interactions between vasopressin and serotonin. And let's just stop for a moment and examine that, because I think this is why it's so confusing. When they say neuroanatomical, what they're talking about is basically they're going to create a three-dimensional representation of this pathway through a series of combined staining results. They're going to be constructing the nature of these molecular innervations within kind of like space itself, and then they're going to be, after doing that, after kind of like establishing the shape of these neurons and the way that they kind of like reach each other, they're going to be looking at the neurochemical interactions. So that is the action of these neurotransmitters kind of between and around the neurons themselves. And then after that, they're going to be trying to make inferences about what's going on inside of the neurons when these neurochemical interactions are occurring. So yeah, the reason why it's so difficult, I think, is because that is complex and confusing, and they kind of hit you with it in a weird way. This first page, this second page, this is entirely methods. These are the imaging methods. These are like three completely different and super complex neural imaging methods. And this is just a long list of like serums and stuff. You're going to forget entirely about it. And then after that, they hit you with the behavioral assay, completely distracts you. Now you're thinking about hamsters in a box. And then after they finish talking about the behavioral assay, they hit you with two of the densest paragraphs I've ever seen in my entire life. These two paragraphs, they summarize the entire neuroanatomical and neurochemical interactions that they found by combining the results of these three different imaging methods. So that is just so hard, and you're already distracted and you've forgotten about the neuroimaging by now. And then after they construct these pathways, they hit you with the behavioral effects. But that makes no sense, because that's what they assayed first. And yeah. And then they get to the discussion, and then it kind of just goes back and forth for a while, and I don't know. I'm going to be going through it chronologically. So let's start with the behavior, and let's leave the neuroanatomy and neurochemistry for a little bit later. So they're conducting 3 series of microinjections, all on the same hamster, and then measuring the hamster's behavior when confronted with 4 different tasks. So the hypothesis, again, is that serotonin inhibits vasopressin, and that vasopressin mediates for aggression. So serotonin stops aggression by stopping vasopressin neurons. To this end, they hypothesize that if they give a hamster fluoxetine, a selective serotonin reuptake inhibitor, it will subdue this aggressive response by increasing the amount of serotonin in the synapse. So yeah, the way they determine the dosage of the SSRI is by measuring, quantifying its effects on the behavior known as flank marking. So flank marking is something that hamsters do. It makes them particularly suited for this type of research, because they have glands that excrete a musk, and when you give them vasopressin, and you give it to them in high amounts, they'll excrete this musk like crazy. They'll actually ignore resident intruders and just run around their cage spraying musk everywhere. And then when you give them a vasopressin antagonist, they won't spray any musk. So, we know that, I guess, vasopressin also, in addition to mediating for aggression, mediates directly for this kind of flank marking behavior. So that's how dosages of SSRIs were determined, by seeing how much the SSRIs dampened flank marking. So let's go over the tasks. First one is resident intruder. They're going to be measuring aggression. Second one is a sexual kind of like motivation and copulation test. They're going to be giving it a female, and then before it ejaculates, they're going to take it out. And they're going to put in task 3, which is a box lined with the odors of other hamsters. They're going to be measuring flank marking behavior here, which again, mediated by vasopressin. And then the fourth task is a really big box that's been broken into 4 quadrants, and they're going to be measuring motor activity by just counting the amount of times that the hamster runs in a circle, like around the box. So let's get to the results of the behavioral section. Figure 4, that's showing us the effect of the SSRI. This first 2 is measuring aggression. We see that it increases the latency to bite and decreases the amount that the hamster bites to 0. We also see that it completely subdues flank marking behavior. Fluoxetine also, these last 2 here, or these first 2 on the bottom, that's kind of the sexual motivation. We see that it somewhat dampens sexual motivation / copulatory behavior. And then this last one is just showing that serotonin doesn't really have much activity on the motor activity, on the motor activity of the hamster. This fifth figure, this is really where kind of like you can visualize the effect of these molecules and this pathway on behavior. This first one, again, it shows us that fluoxetine greatly increases the latency to bite. And this second one, it shows us that when you inject a hamster with vasopressin, it significantly decreases the latency to bite. And then this other bar chart, we can see that it significantly increases the amount of times that bites the other hamster. So, from this, we can infer that vasopressin does, in fact, mediate for aggression. That if you give a hamster vasopressin, it becomes more aggressive. And if you give a hamster an SSRI and increase serotonin, it becomes less aggressive and subdues that aggressive response. So that is the behavior, and those are the behavioral results. Okay, so onto the neurobiology of it all. I think that this part is really interesting, and it's a shame that kind of gets lost in the research, but let's go over it. So remind yourself of the hypothesis. We're talking about how serotonin, by inhibiting vasopressin, leads to subdued aggressive behavior. So researchers, they wanted to know the neuroanatomical and neurochemical interactions that exist within this inhibitory pathway, or within this just kind of net pathway itself. And so the way that they go about doing this is by taking 3 neuroimaging assays and basically combining the results from all of them to infer a lot about the kind of the shape and chemistry of these pathways. So let's start with the neuroanatomy. So what we're going to be doing is visualizing this pathway and on a molecular level within 3 dimensions. So we're going to be thinking about how these neurons exist in physical relation to one another, and kind of how they exist on a 3-dimensional plane. So I'd like to take your attention to figure 3. What we're looking at is vasopressin neurons in red and serotonin neurons in green. At the top, and this is within a slice of the anterior hypothalamus. So we're looking at the anterior hypothalamus. At the top right, that big glob with a star on it, that's a vasopressin neuron. You can clearly see the body as well, the axons and the dendrites. And then we can see that there are no serotonin neurons present. And so let's go back to that idea of depth, that dimensionality. Let's assume that this image is falling away from us, that we're kind of like looking into it and it moves back. It has depth to it. And with this understanding, we can conceptualize kind of the nature of this innervation. In that at this level, this plane of the anterior hypothalamus, we have vasopressin neurons, and then serotonin neurons are coming from a peripheral secondary location. They're innervating up and onto these vasopressin neurons in this plane of the anterior hypothalamus. So considering the larger cellular cut and structure of this region, let's move to figure 2. So figure 2 is immunohistochemistry. We should all be very familiar with how this works. And so what we're looking at is the kind of the same plane. We're a bit more zoomed out now, and kind of it's a bit more two-dimensional. But we are evaluating in the top image vasopressin neurons, and in the second image serotonin neurons. What does this tell us? So the top image clearly shows that there are vasopressin neuron bodies in the anterior hypothalamus as well as around the anterior hypothalamus. We can see them. There's a very clear one on the right here. Anyways, the second image, that shows us there are no neuron bodies of serotonin cells in the anterior hypothalamus. However, there is a massive dense plexus of neuroreactive or immunoreactive fibers. So what does that mean? It means that vasopressin neuron bodies are located in and around the anterior hypothalamus, and that serotonin neurons, they heavily innervate this region from a secondary location. But the innervation is huge. It's massive. There's immunoreactivity everywhere. And so let's think now about the action of the individual neurotransmitters in this pathway. So I'd like to draw your attention to figure 1. This is audio radiography process I don't believe we've talked about in class. It was very interesting. They didn't do a very good job labeling these. Just like write it down if you haven't. What A is V1A receptors. What B is 5-HT1B receptors. And so the way this works is researchers take a radioactive molecule, they bind it to a ligand, that ligand binds to a protein. Once that ligand is bound to a protein, you have this radioactive molecule that is now basically tethered to whatever protein you want to study. And by using a particle detector, you can create a map of basically all of these points of radioactive emission. So what we're looking at in both of these images is every single one of these black dots is a receptor. What does this tell us? So B tells us that there is a very dense concentration of serotonin 1B receptors in all around the anterior hypothalamus in a very homogeneous way. What A shows us is that there are very dense kind of clusters of V1A neurons. In that box right there, that's denoting the anterior hypothalamus. And the important part of this is when we combine these two images together, we can understand that V1A receptors and HT1B receptors, while they're occurring in the same location, they're co-localized. So they're found right next to each other. And so what do researchers draw from these findings? Well, they posit that serotonin could potentially inhibit vasopressin neurons in 2 ways. One is a indirect way and one is a direct way, a direct inhibition. So let's first talk about the direct inhibition. So serotonin neurons innervate vasopressin neurons at the level of the anterior hypothalamus. We know that the vasopressin neurons are going to innervate a secondary neuron, that they have to kind of like send this excitation forward, that another population is going to basically carry this signal. So we also know that there are secondary neurons probably in the anterior hypothalamus that are being activated by vasopressin. And so when we consider co-localization for a moment, we can start to understand that these secondary neurons that vasopressin neurons synapse onto might co-express both serotonin receptors and vasopressin receptors. And so a direct inhibition can occur as serotonin neurons release serotonin, which binds onto these neurons that have this dual expression, and inhibit the effect of vasopressin. And this would inhibit vasopressin after vasopressin has been expressed by the neuron. This is probably this because it's also occurring at the axons of vasopressin neurons. Researchers also infer that serotonin, in addition to kind of like this binding to co-localized proteins, could also be binding to the axons of vasopressin neurons themselves and inhibiting vasopressin expression that way. And so now let's get on to the indirect mode of inhibition. And we're going to kind of delve a little bit more into the molecular inhibition of the neurons. So 5-HT1B receptors have been implicated by previous research in inhibiting the expression of CAMP. So yeah, when serotonin binds, CMP goes down. Vasopressin neurons, on the other hand, well, they have been associated with high levels of CAMP, and V1A activation has also been associated with higher levels of cyclic AMP. So the idea for indirect inhibition is that serotonin neurons innervate onto the vasopressin neuron bodies that are around the anterior hypothalamus and the NC and the MSON. And then by inhibiting the expression of cyclic AMP, they inhibit vasopressin release and vasopressin synthesis. So we think about this kind of like interaction between 5-HT and cyclic AMP. We can also kind of infer the activity going on in this like postsynaptic co-localized receptor neuron, in that like the activity of vasopressin increases CAMP and the activity of serotonin decreases cyclic AMP, or this activity of serotonin 1B receptors decreases cyclic AMP, and that likely the metabotropic activity of HT1B receptors is stronger than V1 receptors, and just kind of acts to stop that expression of cyclic AMP due to V1 in the post-synaptic neurons. So yeah, I hope that explains kind of like the neuroanatomy and neurochemistry of what's going on. I guess we can move on. There's just a few like small things at the end of the paper I'd like to talk about. The first one is this idea of a serenic. I think it might be a little silly. It's just fluoxetine. They're basically suggesting that because we know the neurochemistry and neuroanatomy of this pathway, and because we can kind of like selectively inhibit aggressive behavior at this level, we could give people who are overly aggressive, who have hyperaggressivity, some sort of SSRI that will kind of exert a greater inhibitory effect over the aggressive behavior of VP, which is cool. And then I like to talk about potential confounds. So there's one really big one. And there's only really one. I mean, there's like three, but there's one that we really have to focus on, and that is the nature of fluoxetine in the brain. So the fluoxetine and SSRI increases the concentration of serotonin, of course. And yeah, we can kind of infer directly what is going on in this pathway because of that. But it's not localized. This increase of serotonin is something that's happening globally. The SSRIs are affecting every single serotonin transmitter in the brain. So everywhere where there is serotonin, there is going to be more. So if there is any possibility of like another pathway that mediates aggression or modulates aggression, that also has that also has serotonin involved in it, that is going to also be kind of overexpressed and overactive. So there is the potential that by globally increasing serotonin, they were just exciting another pathway that was kind of like mediating for this behavior. So yeah, that is the video. I hope you guys enjoyed.